chit-chatting about your projects. Michelle, you have a book for us, right?
Yes, I do.
Okay, cool. Let me just wait for the live stream to be up. Looking good. Here we go. Welcome to Neuroccino on Monday morning. This is an online journal club where everyone is welcome to bring a piece of science they get excited about. That could be a paper, that could be a book, which is a new category, and briefly present it, and then we discuss it to the best of our knowledge. Today we have the first book presentation and one paper as far as I can see from the chat. So without further ado, I hand over to Michelle. I promised you to go first, and you can present your book to us. Thank you.
Hello. I'm going to present you this book that I took 2 years to read. It's Myelin, the Brain Supercharger from Bernard Zalk and Florence Rosier. You can find it into our chat in the PDF version. I will share my screen so I can show you some of this. There you go. So now you can see the book with this beautiful cover. So if you don't know, the myelin is actually this little sheet that is wrapping the axon and increasing the speed conductivity, as well as working as an isolant for the electric charge that will go through the wire. Otherwise, there will be a total mess. All the wires will share all like their information, then we will not be able to communicate correctly in our brain. This book is the life achievement of Bernard Zalk that he nicely dedicated to me because he's a friend from ICM in Paris, and is jam-packed with very interesting information about glial cells, not only myelin, but also glial cells. So I took some note in my printed version. I'll try to find it inside the PDF. But if you start with the first chapter, I think it can just do this. There you go. Here you have like the real numbers that correspond to the number of neurons in the human brain and the number of synapses in the human brain and the number of synaptic signals that happen every second. So if you look for a reference for these big numbers that are usually kind of fancy or help to teach students about like the dimension and the complexity of the human brain, here you have a book that actually, with a true scientist that is doing histology that been doing histology for his entire career, has been estimating and bringing inside his book with accuracy. So those numbers are not exaggerated, like what you can find in the press. Those are just sample of informations that I found interesting. You have all the history of like how we discovered myelin and glial cells, which is important because I didn't know about like the history of this, and we're all very neurocentric. And then other important information that you learn is that the proportion of glial cell to neural cell in the brain is not the same according to species. So for example, if you take the Drosophila, like the proportion of glial cells has been determined to be between 30 and 35% of all cells in the nervous system. If you take the rodent, the proportion is 65 to 70% of all the cells, with only 30 to 35% being neurons. And in human, it's 90% of glial cells and only 10% of neurons. And so like you see how you can put it into an evolutionary perspective that like the glial cell is really developing all the potentials that exist in the human brain. In the introduction as well, I remember I really like the beginning of the book in the kind of way the authors explain to you what is the main role of glee, which is to increase and finely tune speed in the brain. So I didn't know, but the flea actually doesn't have glial cells. So it doesn't, it's only unmyelinated axons. And so this myelination of the axon is principally due to the fact that you need to increase speed to go from longer distance in the brain. So that's an evolutionary strategy that happen in the brain. So if you look at very tiny brains, usually they don't have myelin. Then here, so you have like, not all glial cells produce myelin. Some others, like astrocytes, are like feeding the neurons and helping with the process of communication between the neurons. And you also have the neural stem cells, which are also glial cells. Those do not necessarily produce myelin, but are part of the glial cells that are nicely introduced in this book. And so why is it important? Well, it's mostly because we live in a neuroscientific world which is very neurocentric. So we're really interested about neurons, are like these little boxes indicating you get about more than 500,000 articles with the word neuron. But if you're interested about glial cell, you have only 100,000. And if you're interested about specific glial cells, these numbers are really dropping. And that's because, it's a new addition. If you look at the history and the study of the glial cell, it's a new addition in the way we look at the working of the brain, and particularly something that is developed inside the book very elegantly with a lot of very interesting examples that are solid histological example, is the link between glial cells and the plasticity of the brain and the development of the brain. So, if that is true, that if you look at the brain on a static perspective, neurons will have maybe the lead on to how things happen. But if you look at how you can modify the functioning of your brain and learning new skills, development or aging, then a glial cell have a crucial interaction with the system. All right, so some other interesting informations that I found there. I know we had the debate for a long time with Steffi, and those are information that you think you sort of know, but it's always good to find a good reference that is like giving the exact numbers. So it's been measured that a myelinated nerve fibers can go from 288 to 432 kilometers per hour. Okay. And so that's also an information that, we always said 300, 350, but here this is like the clear estimation of that speed. And this is derived from the equation that modern nerve impulse condition velocity that led to the Nobel Prize in Physiology of Medicine for Hodgkin and Huxley in 1963. Books, got all those information just in books. Just stop reading papers. Just kidding. I'll drop the example, but that was just to give you an idea of why it is really interesting. Then like the rest of the book is going like higher and higher in terms of complexity, going to the fine structure of the myelin and the way everything like the way the oligodendrocytes that produce the myelin actually communicate and exchange. And the books finish with like examples of pathology of the myelin, the mechanism, and what are the latest development in terms of research to compensate from for pathology of the myelin. I think that's about it. Don't want to give you too many examples. I want to encourage you to read the book. You found the link in our chat. If you are interested, you can drop me an email directly and I can send you the book by email. And most importantly, if you're very interested, you should buy the book because Bernard and Florence spent a long time writing it. Thank you very much.
Thank you, Michelle. That was absolutely lovely. Before I open up for questions, just two points of clarification, really. So I'm glad to see we finally get a reference for our speed of conduction. 300 miles, 480 kilometers. So not too far off.
Yeah, we're right in the middle, but it was good to have like the who said it first, where does it come from, and now we know it's coming from this Nobel Prize in 1962, and we have the exact value and the equation that relate axonal diameter with myelin sheath and speed of conduction.
Having a Nobel Prize as a reference does never hurt, I guess.
Yeah. That's the best that can be.
The other thing I wanted to quickly do is before we open up for questions is that you tell us what glial cells actually are. Because you kind of dived right into it.
Oh, yeah.
What are those cells? How many types do we have? What do they do?
So you got like
the expert by proxy. So
but the book is about the myelin, and the myelin is supported and fed by oligodendrocyte in the brain. Schwann cells are outside the brain, peripheral system. And oligodendrocyte are glial cells. Glial cells, by definition, are non-neuronal cells in the brain. And they include like microglia, which is absolutely awesome and can like travel from one area of the brain to another area of the brain by itself just to clean it up. Ependymial cells, which are like the stem cells that you can find a little bit everywhere in the brain, but mostly at the level of the ventricles, and they have like a cilia, and they can like move when they're not transformed the CSF. And after a stroke, apparently they can transform in neurons to provide support to the brain. I'm just amazed by that. You got astrocytes. Astrocytes not only feed the neurons, but they also modulate the blood flow, and they can modulate apparently neurotransmitters by reuptaking and releasing the precursors or neurotransmitters, if I remember well. I think that's about it. I'm not an expert of glial cells, of course, even though I read the book. Another thing that I found fascinating in the book is throughout the end, but like there is a clear demonstration and people have been able to show that oligodendrocyte change the myelin conduction to synchronize the inputs in one region. So let's say you have a region A and communicate with B, C, and D. And to trigger the action, which would be moving your finger, you need to have B, C, and D that arrive at the same time onto region A with electrical afferences. This is some very fine tuning when you look at the speed of conduction and the amount of electric charges that will arrive at the same time. And this is very finely tuned to be perfectly synchronized by the oligodendrocytes, influencing on the myelin that will modulate the speed of conduction, that will make your signal arriving at the same time in the region. And yeah, this is one of those fascinating biological evolutions that made like the system of the brain so finely tuned.
Lovely. Any questions? Yes.
Thank you for the book, sir. I just wanted to ask you, do we have information about similar proportion of glial cells or their functioning in non-human primates? Because if we are thinking about maybe evolutionary contribution of
that's a very good question. So it is not in the book, but I'm sure somebody must have counted that, and if not, they should, because that's a very interesting question. And having like different primates will give us a possibility to really infer that is this like something, this 90% of glial cells, is this something that is specific to primates, or is it something that happened earlier in the phylogenetical tree? And it's a good question. The same thing is like, so you have a lot of myelin to compensate for the long distance that have been traveled by connections. But I didn't see, and it is not reported in the book, whether anybody did a correlation between the proportion of glial cells, or particularly oligodendrocytes, and the size of the brain. And there should be a relationship there. And if the relationship is linear, it may be terribly boring. If the relationship is non-linear, that means that there is something even more than just compensating for size.
Thank you. Yeah.
Leah, you had a question as well.
Yes. Thanks for presenting. Now I'm curious about the book, and I wanted to ask about the structure of the book. So it's more based on topics. You said at the beginning there is an introduction and then some pathology examples, or is it more historical? Because usually in these books that are in the big not review, but state of the art, sometimes you have a lot of reference to articles that the findings that come up. How is this book organized?
All right, yeah, sorry, I went quite fast on this, and I didn't really have in mind a good structure to explain the book, but the structure is the following. So first, and it's a smart structure actually to write a book. So if you want to write a book, I found this pretty good. First chapter is to tell you why myelin is so interesting. So it's so funny example, the brain of the brain of the flea, or it just doesn't have myelin, and why do we have myelin? And there are all these funny things about you have to react fast, and how much time would it take if you didn't have myelin. So the first book is lay reader. The first chapter is lay reader, getting you into why it is important. Then the next chapter, how did we discover that, and why is it important, and how did we understand the functioning of it, which expand not only onto myelin and oligodendrocyte, but cover also other glial cells, because those discoveries happen at the same time and were made by the same people. Okay. Then it dived directly onto very advanced measurement and biological things that happen with the myelin at a level of structure and interaction that I never thought about. So I was kind of whoa, this is really advanced, which I think reflect really all the latest work from the authors. And it finished up with all the relationship with behavior at a bigger level, where they nicely cite my work, but I really liked it. And the pathologies, and this myelin pathologies, and what are the current treatments that can happen. It's about and also reference in the end. So it's well referenced. You have good examples and original references. By original, I mean who was the first for every statement that I made in the book, and that's why it's so valuable for us, because that helps us to not only think differently and embrace really the vision from somebody who spent his life working on this, but also if we'd like to take the train of the glial cell with those authors, now we have the good references and the logic and the credential to do so, and that's why books are so important.
Okay, thanks.
Lovely, thank you very much. Any other questions, comments?
Yes. Me?
Yes, Evie.
Yeah. So Michel, we discussed not a long time ago about the parallel between evolution, so between phylogeny and ontology. So I don't know if he's discussing about that, as he has a chapter about evolution and a chapter about development.
No, I missed it, or like, yeah, I didn't see any link being made here, and you know, the onto like the philo-onto hypothesis.
Yeah, philo-ontogenic.
So you know, there are some links showing it, but like a lot of it has been debunked, particularly like the Princep work from Echo have been debunked, and they showed that there was some problems there. And there are also papers showing that there is a link, but you know, I think this is really an hypothesis that needs to be investigated properly with current technologies and nice evolutionary maps. And so I guess like a good first step would be to do the first evolutionary brain map. So yeah, no, it was not really discussed.
One more question that just came up in the chat, and you might actually know the answer to it, is if Einstein had more myelin.
Oh, that's a very good question.
No, it was a joke. It was a joke.
All right, so we don't know if Einstein had more myelin. We don't know if Einstein had more myelin, but part of his brain has been sent to Marianne Diamond, who worked on enriched environment and thickening of the cortex. And we know he had more glial cells, particularly astrocytes. And so if you use a lot your brain, this paper from Yanni Fessef, for example, if you play video game and you play all the time a different circuit, or you play all the time the same circuit, you'll have differences that happen very quick into your hippocampus. If you play all the time the same circuit, and this is because you map those this circuit into your brain, and those differences that happen in just 2 hours are related to an increase of the size of the astrocytes. Now, and they know it because they did it not only in humans, they did it in mice in parallel, using kind of a pool maze paradigm. So in the case of Einstein, that's not only the size of his astrocytes, that's also the number of them inside the cortex that were more important. That was in his parietal lobe, and it's been linked with his fantastic ability of abstraction, of projecting himself visually inside the problem and finding those metaphor or conceptual representation of the problem that help him debunk fundamental physical questions.
Thank you very much. Victor, yes.
Hey, good morning. I just had a question that came to my mind right now. It was, did they talk about in the book about the way we have today to measure how much people have myelin, how much myelinated their axons are in vivo, and what kind of technique we have today to do that? And to
No, this is not something that is covered in the book, and it's mostly because the authors come from the histological wet science world and take the result from MRI just for granted and don't question their validity, because this is not their field. Like, I will not question the validity of the histological report. That's just as simple as that. So they report findings, but there is no chapter onto advanced in vivo measurement of myelin and how we would do it. Okay. But there are some examples of application, particularly in the neurodevelopment, plasticity, and pathology that are derived from MRIs that quantify the myelin. They just don't discuss the methods. They just discuss the results.
Thank you.
Bye, Abe.
Hi, Abby. Thanks for coming. Well, that was an exciting discussion. If there are no further questions from what I can see, we move on to the next paper. Meanwhile, I'm going to go on YouTube.
Okay, so this is a review on comparative neuroanatomy, and it was published at the end of January, but I had the chance of read it now, and it was from Mars, Jacobi, and Rushworth, I think. And I really like it for the how much it was clear in the explanation, and the figures are really informative. Indeed, I gonna explain the review starting from the figures. At first, of course, so not first the title. So a common space for approach to comparative neuroscience. So they are trying to find which is the best strategy to compare different species, but also different modalities, because now we are able to have a lot of information, not about the brain, not just from one methodology, but to several. And so questions arise to how to combine all these knowledge and which is the best approach. So here there is an explanation of all the possible ways that you can do comparisons, find similarities, or what is different between the different species, and so to build this new framework, they talk about this vertical translation and horizontal translation. So in the vertical translation, you can see here, you have a comparison between different modalities, but within the same species. Otherwise, when we talk about horizontal translation, we have the comparison of the same modalities, but different species, so across the species. And then you can compare these approaches. So, for example, you can study cortical folding together with cortical myelin, as we talk about, or tractography or gene expression in the same species, or translate. So the first approach is use the same modalities across the species to make a comparison. So let's imagine in this radar graph that you have different species, and you can have the same modality. For example, here in yellow, let's imagine like the default mode network study in the different species. In green, the tractography investigation. And then you can have also other modalities in some species that you don't have in humans. So for example, in blue tracers methods. So this is all the information that you can have, and you have to put in together. And one way to do it is using an abstract approach, so extracting features that can help in the comparison of the different species. So they suggest principal components, space embedding, or just measures like the strength of connectivity, something that can be like an abstract feature that you can compare, and so bringing all the information in a common space that is comparable. Because usually, as we know, there are differences about the brains across the species, and about the size, the regional expansion, the increased lateralization, new projections, or change in the connectivity strength. So all this information that brings some difficulties in the comparison, but it's also the interesting part. So here a way to extract this information and use it in the informative way. And to do that, they suggested to use, they say, 3 ingredients. So the first one is the multimodal information. The second one is to focus on local data, so local information. And the third ingredient is the abstract features space. So a features-based comparison in a common space. So something that is abstract, and then you can overcome these differences that we have in the brains of across species, but you're able to make an informative comparison. And so, as we have said, vertical translation is same species and different modalities. So here vertically, for example, if we focus on the human brain, you can see that here we have the T1, T2 weighted maps that can resemble the gene expression in different cortex layers. But here you see that each map seems to give like a detail of these of the T1, T2 weighted images. But if you do a principal component of the different layer gene expression, you have actually something that is comparable to the first one. So a comparison across modalities and using these new embedding space, so a feature-based to make a comparison. Or you can go not vertically, but horizontally, and you can try to compare different species in the same modality. In the same modality, and you can find like a correspondence, for example, between some cortex layer, and use that for guide the registration and the comparison. You can and so like unfolding a comparison between different species, and sometimes you do like correlations of correlations. So is to what I understood is a way to find both the similarities and differences, and you can make, for example, this map of unpredictability. So how much is the difference across the species? And then you can also combine these different approaches. There was here. So you can do both horizontal and vertical translations. So for example, when you have the same modalities across species, you can use it for a comparison, but also you create like a prediction and see how much is the goodness of this prediction, and you can use this registration then can be applied to other modalities. For example, the investigation of the arcuate fascicular projections, how it's different across the species, if it is more vertical in primates, how much is the expansion in humans. And then you can put everything so in the same space and see here in humans how much is the prediction in the human space, and how is the human actual arcuate projections. And so here you can make an unbiased comparison because you bring all the information together in the same space. Or when you have, for example, tracers information that are available only in primates or other species, but not humans. And you can use like a method that you have in both species, such as like tractography. you can use this registration to then make informative information also in humans of some tracers information that you have here just in the macaque. And yes, this can be made also for other, I think it's this, no, there was also an example in the test about the food mold network that was proved to be detected across different species, but the involvement of the areas are actually different. So you can make all these comparisons that I imagine are really never done, but I imagine that are like challenging, but this is a framework how these approaches could be done. And so I think, yes, that's it. And of course, there are a lot of important references, examples that are really well explained. And yes, this was the part about the food mold network. And so this is what I understood from the article. If you have any questions.
Thank you, Leah. That is a very interesting paper, quite clever. Are there questions? Patrick.
Yeah, thanks for presenting this. Really, really cool. For some reason, I haven't got this paper yet. I even asked Roger about it on ResearchGate, but he hasn't answered. So what I was wondering is, do they propose different common space approaches for the different modalities? Or is it always along the same lines? Because, for instance, like some while ago, I think 2018, Roger published this this white matter blueprint paper as one way to establish a common space between species, but this is potentially not feasible in other modalities, right? So how did they solve this?
Maybe Michelle and Stephanie have a better answer. Actually, I don't know. Yeah, I
So, oh, sorry. Yeah, for the purpose of clarity. So I'm writing a book with Roger, so we speak actually very often about these things. And one of the key is the multimodality of the data. So if you have enough different measurement for each of your species, and those measure, the key really is a comparability between your different samples. And if you do the same measure if you do different measures that are the same between your samples, then you can concatenate it into the same space. For example, if you take macaque and humans and you do diffusion, myelin, electrophysiology, and other things, and you concatenate enough data together, then you'll be able to bring them into the common space, which will be based on the similarity in the measurement. Does that make sense?
Yeah, but from a technical standpoint, does this mean that the data should be not only similar in terms of the modalities acquired, but also in terms of the dimensionality? Or
no, it's not necessary. So you can concatenate anything if you have only one point of if you have like one point of similarity. This point of similarity might be like Roger has been doing. So you use an atlas of the connections. So you compare IFOV and IFOV between two species, archaeid and archaeid between two species. And so you can line them up together. Another way is to do it based, and that is what it suggests, multimodality, which will be completely data-driven. It's just you acquire the same modality in all your sample, and then that will be your point of comparison between your participants. So it will not be based on space, it will be based on profile on these different point of measure. And the more you have, the more accurate will be your space and your distance between your sample.
But this still doesn't really answer for me, like, what is the method behind it? So is it an embedding technique? Is it dimension reduction first and then looking at similar, like, reduced components? Or
it's a good, well, it is a review, so it's not an original paper. I guess, no, I didn't read that paper, but there are like different ways to do it. And what they suggest is PCA probably to cancel out the noise and focus on the main, driving factors. And then the embedding, and they speak about embedding, you have many different approach for the embedding, which here, I don't think they take like a solid standpoint or stance onto one method compared to another. It's more the idea of doing it rather than how to do it in terms of like practical method and algorithm. But we'll get there, I think, one day when we have enough data, hopefully.
Yeah,
yes,
really cool. Nice.
Like looking through the papers, like here in the horizontal translation, you can find so the technique, a little bit like some tips you were looking for the comparison. So as the first approach, they suggested to use homologous spatial landmarks. So you, for example, a cortical, the same, such as the primary spulci, a cortical sheet folding that you can have both in macaque and humans, or you can use this approach also from like starting from the cingulate and parietal cortex. So there are also the reference, but use landmarks. Here there is this blueprint connectivity that maybe you were talking about, where you created this connectivity fingerprint. This is like a matrix, cortex, white matter connectivity. So you have this big matrix that here's your connectivity blueprint. Otherwise, if you want to focus on the embedding, as we were saying, here we move on the vertical comparison, and you can use both the principal component, but also other methods. And here it's also, of course, for the embeddings, you will use features. And here there are some examples about the connectivity strength, if you want to see what the article said.
But I mean, the main question here was if they suggest somehow to try and use all these different embedding techniques or different comparison techniques with regards to the individual modality, or if they can kind of propose like one generalizable solution that fits it all.
No, they don't say that. They're not proposing like they don't make like a classification, so which is the best method or not. They just report all the methods for the purpose of this vertical and horizontal comparison. No, they don't say this is better or no. Sometimes they say this was like at the beginning and there was an evolution, so now we can compare it better this way. But they just present all the methods and the specific features of all the methods. So they don't propose the better one, which is the best one.
Yeah, thanks for clarification. That makes me want to read it now.
Mission accomplished. Right, are there any other questions on this paper? Not that I can see at the moment. Nothing over on YouTube. Well, with that, thank you very much for coming today. It was lovely to have your company this morning. And we shall see you next week again for more exciting science. Have a good week, everyone. Bye.